Hi everyone, my name is Karishma Gangwani. I am a computational scientist at St. Jude Children's Research Hospital, and we have developed the web-based Medulloblastoma meta-analysis portal for an in-depth analysis across three large clinical trials of medulloblastoma, the most prevalent malignant brain tumor in children. And you can find more information about this disease online and in our published studies, links for which are provided in the description below. And the link to access this portal is also provided in the description. And that link is proteinpage.stjude.org/mbmeta. Now, this link houses the manuscript figures, both the main meta-analysis manuscript as well as the web portal manuscript. But we will just use this to launch the portal. So to launch the portal, all you have to do is go to this link and then click on this button called Launch Portal. And it should take you right to this page where you will see this Welcome to the Medulloblastoma Meta-Analysis portal highlighted on the front page. Now, this portal is in itself a major effort for enhancing data sharing efforts with the neuro-oncology community. Once you're inside this portal and you see this About page, you will also see some information about this portal. There are 898 patients across three major clinical trials of medulloblastoma in this, and two of these trials are COG trials. And in this portal, users can stratify patients based on various clinical, demographic, and genomic level attributes to assess risks and outcomes and make informed decisions about therapy, or basically use it as a discovery tool. So in this tutorial, we will cover the data dictionary and how to access the data dictionary to perform, basic analyses. And then we will also look at summary plots like bar charts, violin plots, and scatter plots. So let's begin with the data dictionary. For that, you have to go to the charts tab, and that's where the data dictionary is housed. And you can see that's the very first button under the charts tab. So if you click on data dictionary button, you should be able to see some of these terms that are housed under these parent terms. So all the variables that are present, for this 898 patient cohort are all housed under this data dictionary. And let's click on one of these. So we can click on demographic, which is the main umbrella under which these three different variables are housed: age at diagnosis, age group, and biological sex. And so all of these dictionary variables have these term informations right next to them. So if you click on the information icon that is shown here, you should be able to see the description for each of those variables. So at any point in the process of analysis, you do not remember what exactly that variable means, you can always go back and look at these definitions. So for age at diagnosis, you can see the description. The title is age at diagnosis, and it is the age at which a patient is diagnosed with the primary disease. So what we can go ahead and do is click on that variable. So each of these variables are clickable. Now this is a very intuitive and user-friendly interface. So let's click on age at diagnosis. And you can see this bar chart is loaded with different buckets showing age ranges from below 5 to more than 11 years old, along with a descriptive statistics or a summary statistics that is loaded below this bar chart. And as you can see, the median age is around 8 years, with the maximum age at diagnosis around somewhere around 22 years. Now we can additionally change these age ranges based on varying bin sizes that you want to create as a user. And you can do that using these setting button, which is shown by the burger menu here. So go ahead and click that, and you can click on the term age at diagnosis, which is within the row customize bins. And you can click on edit and go to varying bin sizes. And in the box where it says bin boundaries, you can create the bins that you want to visualize. So I have selected 3, 6, 12, and 16, and always remember to hit the enter on your keyboard so that it gets applied. So the bins that you will see on the bar chart are now lower than 6, 3 to 6 years old, 6 to 12, and 12 to 16, and then an age group which is 16 years or older. And so click apply to see this effect take place. And now you can see that there are 4 bins that are created based on the numbers that I provided in that bin boundary box. So this creates these bins and updates the visualization. And we can further play around with the settings here to get the perfect visualization that we need. So now I want to change the orientation, and I'm going to go and click on the vertical orientation so that this bar chart is now vertical, as you can see. And we can see that the maximum number of samples are represented by this 6 to 12 years of age group, and they fall into this bar chart. Now, the other thing that we can do is we can stratify this further based on other variables. Now, there are 4 major groups in medulloblastoma, that is the Wind, sonic hedgehog, group 3, and group 4. And those are further stratified into 12 different subgroups based on their molecular biology. And so we are going to overlay that with molecular subgroups. So, like I said, those are 12 different subgroups, and that is what is loaded here. So you can see that in the legend, we can see Wind, Sonic Hedgehog 1, 3, 4, and then we can also see group 3, group 4 are combined into 8 different subgroups. And all of them are overlaid over this age at diagnosis. And now you can see on these bar charts that are loaded, there are some asterisks. And seems like everything seems significantly associated with that age group. But in order to account for type 1 errors, we have also added multiple testing correction. In this case, we are using the Bonferroni. So if I go ahead and click that, some of those asterisks will disappear. Now, this correction helps to control the significance value by adjusting for it. And so once selected, you can see the actual number of asterisks have now reduced. And so you can see the actual real effect, or if there's any real association for that subgroup or molecular group with the age group, which is also represented by this 2 into 2 association test, which uses Fisher's exact test. And so you can see the p-values on the right of this bar chart in a table form. Now, let's look at this proportionally to get a better picture of what these associations are. So as you can see, from age group 3 to 6, the Wind and then subgroup 3 from group 3 and group 4, and subgroup 8 from group 3 and group 4 are strongly associated with ages 3 to 6. For ages 6 to 12, the Wind and the Sonic Hedgehog 3 are strongly associated with that age group. Whereas for ages more than 16, it is the Sonic Hedgehog 4. And in this way, you can draw inferences and conclusions to get a basic idea of what's present in the cohort and use this for your analysis. Now, the same data can also be visualized as a continuous distribution in the Violin plot. So to do that, you can click on this Violin tab, and you will see a chart rendered with multiple colored violins, which is displayed here. And this is again on the left is the molecular subgroups that are loaded, and each of these subgroups, these categories, can be hovered on to view the descriptive statistics of that category. And on the y-axis is your age at diagnosis. So I'm going to go ahead and make this chart vertical because it's easier to see it that way. And then I'm going to reduce the length and the thickness of this chart by using the plot length and the plot thickness function in the settings. Now again, this control panel or this settings panel behind this burger menu is kind of more or less similar across the charts, but the settings within it that might be differing from one chart to another. So now I have reoriented this and adjusted the size, and you can see that all of these violins now fit into a frame. And now I can, do further analysis on that. So if I hover over the wind, I can see that there are total 131 samples, and the median age for patients in the wind subgroup is around 10 years old, and the maximum being 20 years old. And that is represented by this violet color. The red lines that you see in the middle of the violins are the medians. So that 9.81 median is what is being shown by that red line in the middle. Now, each of these violins also have sample level data plotted on top of it, and we can see that using this data symbol, right? Currently, it is shown by ticks, which is very faint and difficult to see, so I can turn that into circles. And now you can see the different samples that are along the age at diagnosis for those particular molecular subgroups. And I can use this brush to drag upon that violin, and two options show up here. Now a user can choose to filter down this age range from 7.6 years to 12.9 years for the wind subgroup, and what that would do is that would take that category and add it as a filter in the filter tab, which gets applied across the cohort. But this is shown in a separate video, so make sure to watch that video. That will also be posted on my channel. And the other option is to list these samples, so I am going to go ahead and click that. And as you can see here, now the samples from that age range of 7.6 to 12.7 years old is loaded in this table here, which I can scroll down and I can get to that sample level data to see what age group it is. And in that way, this violin plot is interactive, and you can see for each different subgroup, you can list the samples and look at the age ranges. Okay, now let's move on to the scatter plot. Now let's see how we can visualize numeric data on a 2D scatter plot. Now from the previous, this violin plot analysis, we are simply going to replace this overlay term with another term which is called as the NMF score. Now what is this NMF score? This NMF score is nothing but a non-negative matrix factorization score. It is a continuum score that can be calculated for these medulloblastoma tumors to help determine the risk of progression and death with this disease. Now, the scale itself reaches from 0 to 1, where 0 represents a lower risk and 1 represents a higher risk. So, to view that on the scale, the 0 to 1 for this, I'm going to go ahead and click on Scatter tab, which is shown right next on the end of the violin plot. And now you can see that on the x-axis, you have age at diagnosis with 604 samples. And then you have an MF score from 0 to 1, with 0 representing a low risk, as I said before, and 1 representing a higher risk. Now, looking at this, there doesn't seem to be any correlation, right, between age at diagnosis and this continuum score. So we can choose a regression line, such as polynomial, to see if there is actually any trend or not. And seems like just for a little bit that, which may not be very significant, but there is a reduction in the risk with increasing age in these patients. So what we can further do is add a color for the molecular subgroup to see if there's any strong associations that kind of stand out. Now, what we can see here is that for the formative subgroups like group 1, 2, 3, which are somewhere over here on the top, they are very close to 1. So they seem to have a higher risk as shown by these NMF scores. Whereas subgroups 5, 6, 7, 8, which are down here, seem to relatively have a lower risk, but they don't seem to have a very strong association with age as such. So this is how you can use this portal to do some initial and basic analysis. And in the next video, we will look at how to perform regression analysis, how to visualize different data types on the matrix plot, visualize samples and methylation data on the methylome t-SNE, and then sample level data visualization on the lollipop, sample view, and methylation array data. So keep an eye out for those videos. Thank you.